Welcome again to the 6.6 biology notes over meiosis and genetic variation. This is part 2 of 2, and our objective in this part is to explain how crossing over during meiosis increases genetic diversity. So we already know that meiosis itself, that random segregation of homologous chromosomes, leads to a lot of genetic diversity. But crossover events can lead to even more diversity. Crossover is the exchange of chromosome segments between homologous chromosomes when they are paired in meiosis 1. So take this example. This is a pair of homologous chromosomes. You can see that there are sister chromatids that are still attached. One side of the black chromosome is identical to the other side. All of the black chromosome, all of the genes on the black chromosome are dominant versions, A, B, and D. On the orange chromosome, though, you have those recessive versions, A, B, and D. Lowercase a, b, and d. Remember, those two chromosomes have the same genes, but they don't necessarily have to have the same versions of those genes. So the black is all dominant, the orange is all recessive. A crossover event causes DNA from one chromosome to be exchanged with another. The legs of these chromosomes can easily be tangled up. And if that happens, sometimes pieces of DNA get flip-flopped from one chromosome to the other. It might seem like this would be a problem. But in reality, as long as you still have all the same genes on those two chromosomes, it doesn't really matter where those genes are or which version is on which chromosome. So I have a new pair over there. I now have some orange exchanged for black, and that's going to affect those genes that are on those chromosomes. I still have the dominant versions of A and B on the left, but now I have a recessive version of D to go with those two dominant alleles. I still have the recessive versions of A and B on the right, but now I have that dominant version of gene D to go with those. So crossover again leads to more genetic variation because you have these new combinations. It's called recombination sometimes because you get these new combinations of genes that result from pieces of DNA flip-flopping back and forth from one homologous chromosome to another. This is going to lead us into our next thing, this genetic linkage, in which genes located close together on the same chromosome tend to be inherited together. If I look at those chromosomes again, on the left, always, dominant versions went with other dominant versions. A, B, and D, capital A, B, and D, all went together all the time. And at the same time, the recessive versions on that orange chromosome always went together all the time. But if crossover occurs, now instead of capital A going with capital D, capital A is going to go with lowercase d. So you have a flip-flop there. Now linkage is telling you how likely those two things are going to stay, how likely two different genes are to separate or stay together. A and B are said to be linked because they're very close together on that chromosome and they're really unlikely to be separated. That crossover event would have to happen in between those two genes in order for them to be separated from each other. So A and B are very, very likely to go together. They are still going together after the crossover that we illustrated. So they're linked. Whereas D is not linked to A or B. it's so far away on that chromosome from A and B that it's actually very likely that it will be separated. So instead of having all the same dominant alleles, capital A, capital B, and capital D going together, now I have that recessive lowercase d going with capital A and capital B, whereas also at the same time, those recessive A and B are going with the dominant capital D. So genes that are located close together again are said to be linked, and they tend to be inherited together. Genes that are far apart, on the other hand, are likely to be separated because of crossover. So we can use these crossover frequencies to create these things called chromosome maps. Based on how often two genes separate and how often they stay together, we can figure out exactly how far apart they are on a chromosome. We're just going to be using generic map units, and 1% is going to be equal to 1 map unit. But we need to be able to map how far apart different genes are on a chromosome. So we're going to have data that looks like this on this slide, and we're going to use these crossover frequencies. All of these are percents, 15%, 2%, 8%. 15% of the time, D and F get separated from each other. So that means that they're 15 map units away from each other on this map that we're going to create. Making one of these maps is really just a logical process. All you have to do is work your way outward towards inward, right? Start working from the outside to the inside. So you need to look for the biggest crossover frequency. So the first number that I'm going to work with here is this 25%, because that's the biggest separation. That's going to be the biggest separation because those two genes, E and F, get separated more often than any other two genes. So I know that my extremes in this map from one end to the other are going to be E and F. And there are 25 map units in between because they get separated 25% of the time. So like I said, you start from outside and work your way in. So the next biggest number, the next biggest crossover frequency is that 17% between G and F. So I know that G is going to be 17 map units away from F. So if I start from F and I work to the left and I go about 17 map units, it's okay if it's not perfect. We're not actually measuring this and we're not going to really worry about that when it's all said and done. But G is going to be 17 map units away from F. And then also, if you look at the information from E to G, there's a separation of 8%. So this distance from E to G is 8 map units. 8 plus 17 gives you that original 25. So you know that you're doing the work right because everything's working out the way it's supposed to work out. So E is 25 map units away from F. G is in between E and F, 8 map units away from E, 17 map units away from F, and I just keep adding pieces of information, keep working in smaller and smaller and smaller. So I've covered this 17%, I've covered this 8%. The only thing left to add in here is the gene that's labeled as D, right? So the next biggest crossover frequency again is probably what I want to use, and that's this 15% between F and D. So if I start from F, oops, let me use a pen here. If I start from F and work my way towards E, about 15 map units over is where that D should be located. So I can put 15 on this line to show that distance. Now, E is also separated from D, and it's separated by a unit of 10. But I'm not going to put that in first. First, I want to go from D to G. I know that this unit here in between D and G, this distance here between D and G, is going to be 2 map units because they're separated by 2% of the time. The rest of the distance between G and E is the same as it was before. It's 8. And so I have a completed map now. Actually, I know the order that those genes go in. I know that E is 8 map units away from G, is 2 map units away from D, is 15 map units away from F. And that's a lot more than I knew before. Right? So these genes are located on a chromosome, and we're not exactly sure which chromosome or where on that chromosome exactly. We just know the distances between the genes. So you can kind of start to put them in order. So it goes E, G, D, F according to the map that we drew. We just as easily could have started on the left with F and worked our way to E through D and G. But our completed map needs to be kind of cleaned up. You go all the way from E to F with those other 2 in between. Let me go back and check again what they were. So it's E, G, D, and F. And those map units respectively are going to be 8, 2, and 15. And again, if you add 8, 2, and 15, it gives you that 25 total. That means that you did it right. You get that map the way you want it to look every time. As long as you start from working with that biggest number and work your way towards the smallest number, you shouldn't have any trouble building one of these maps.